We can all agree that most microbes live in harmony with their host. Many microbes aid with digestion, building, and repairing. However, diseases tend to emerge when microbes evolve to become pathogenic, or when microbes native to one host enter a new host. In fact, human interference with the environment usually results in these abnormalities. This video will look at how HIV originated and entered the human population, along with how some people have developed an immunity to the virus. The human immunodeficiency virus belongs to a subclass of retroviruses known as lentiviruses. Lentiviruses are known to be a slow virus, leaving a long interval between the initial infection and the onset of symptoms, allowing the infected to spread the disease without being aware of it. The only other place where lentiviruses are seen are in non-human primates with SIV. The simian immunodeficiency virus is only known to be problematic when it leaves the original host, the primate, and enters a human, a host it's not supposed to be in. The most prevalent theory of how HIV came to be is believed to be zoonosis, also known as human interaction with animals. Human interaction with animals includes anything between hunting, eating, and domesticating animals. In this case, these animals are non-human primates. Of the two strains of HIV found in the world, HIV-1 is the major strain infecting humans, and it is closely related to the SIV strain present in the common chimp and gorilla. During the bushmeat trade, or the hunting of primates in West and Central Africa, many humans were exposed to the blood of non-human primates. The exposure to the SIV strain was actually seen in the DNA of the hunters. A very high proportion of HIV-positive people in rural villages in Cameroon report contact with wild animals and primates. This includes eating, hunting, and keeping as pets. If you look at the same 14 villages in Cameroon, it is revealed that most of the HIV-positive people had the common strain of HIV in the region. 10.4% of the population possessed another strain whose genomes weren't even described, proving that the diversity of HIV was commonplace in rural villages with frequent contact with non-human primates. So how did a disease localized to West and Central Africa go on to infect the rest of the continent? After all, no city in the region had over 10,000 people in it, and the villages were spread apart. Well, a lot of it has to do with African history. Colonialists arrived and created densely populated cities while establishing sex trades, allowing for the virus to flourish. High rates of labor migration eventually led to 35% of truck drivers to be tested positive for HIV, and the low status of women led to 85% of Nairobi's sex workers to be infected by 1986. By the late 80s, South Africa and Sub-Saharan Africa were facing an epidemic. So far, we have learned how HIV evolved from a harmless chimp microbe to a problematic human epidemic. Now, HIV and AIDS have become a worldwide pandemic, causing 36.7 million people to be infected by it globally by the end of 2015. By looking at the genetics of the virus along with the diversity of whom it affects, the immunity to HIV can be analyzed. When CCR5, a protein, is on the surface of an immune cell, HIV is able to latch onto and infect the cell. When it is not, the cell's door is effectively closed to HIV. The mutation in the CCR5 gene is called delta 32, and some scientists believe that the mutation has been inherited from ancestors who survived the massive bubonic plague in Europe centuries ago. A 2005 report indicated that 1% of people descended from Northern Europe are virtually immune to AIDS. Unfortunately, for most hosts, the virus is able to latch onto the protein. Once the protein is attached, it acts like any other virus. The HIV envelope and the cell fuse together, allowing HIV to enter the cell. Inside the cell, HIV uses an enzyme, reverse transcriptase, to convert its genetic material, HIV RNA, into HIV DNA that's compatible with the cell. This conversion to DNA allows for the virus to enter the cell's nucleus and combine with the cell's DNA. Inside the cell, HIV releases integrase, another cell enzyme. The virus uses integrase to insert its viral DNA into the DNA of the cell. Once integrated, the cell begins to produce viruses, allowing them to infect other cells and take over the body. However, much of the serious illnesses seen in people with HIV in the UK now is no longer directly caused by HIV itself. Some illnesses are seen more frequently in people with HIV than in the general population. These include liver disease caused by hepatitis viruses A, B, or C, tuberculosis, heart disease, and certain cancers. Many doctors now believe that provided a person with HIV receives effective treatment before their immune system has been severely damaged by the virus, and if they take their drugs as prescribed, they can live a normal and even long lifespan.